Okay, I'm gonna share my screen, share, and okay, minimize this. I think we're good to go. Let me hide this one as well. Hide floating meeting controls. Okay. Let's see. Okay. All right. Let me do that again. All right. Good afternoon, everybody. And today is my turn to, let me just double check one more thing. Okay. Yeah, it's being recorded as the, okay, great. All right. Yeah, so today I will be providing a quick update on our SCRI USDA funded project on coffee leaf rust control. We have been doing this for almost 2 years now, and I want to acknowledge my co-authors on this research project. Lily was my former graduate student who just finished her master's degree with me last December in 2023. Majority of the data you saw here were part of her thesis research work. And Julie was the coordinator and also helped with working coordination in terms of the location and also application of certain treatments. And Dr. Zhiqiang Zhang was helping with most of the data collection with Lily and also Nina, who is my new graduate student. We'll continue working on this project since January earlier this year. So okay, let me see. Okay, next page. Okay, all right. So it has been several years. I think we do not have to really spend too much time to introduce coffee leaf rust, their biology and their damage, and or even their symptoms. But it is a very critical and important pathogen or pest problem that we need to manage in in our coffee industry. And like I said, early stage symptoms and late stage symptoms. I'm sure by now, most people who are interested in listening to the seminars probably are very much aware of these symptoms already, especially for our coffee growers here in Hawaii, right? But then I just want to briefly introduce our R4 program. Depending on when Julie's webinar is played either before mine or after mine, today also, you might have more information. You might have already having more information about our R4 program. So we are a four-man team, and me as the PI and Julie and James and Ning as the staff members of the program. They have been working on a lot of important plant pest management, especially recent years, the coffee leaf rust, and we are working to actually get the special labels for some of those systemic fungicides to be labeled for people here to use in Hawaii legally. Yeah, and so here is the quick update on this particular research project. This is the USDA SCRI funded project, and we have, as proposed, we have been doing this project for about 2 years now. So year one, from June 2022 to May 2023, completed, and year two was started from June 2023. It will be completed by May 2024. So we have data for year two up until January last month, and my student will be going to Kona Hill again to take the February data, which I don't have apparently yet. But we are mainly looking at systemic fungicides and biological control products in this project. So like I said, the year two is the exact same repeat as year one, but we proposed that for several reasons. The most important reason is that we want to be able to provide a very concrete, very solid research-based recommendation for farmers to actually be able to use certain treatment to deal with coffee leaf rust. And that's the key information. That's the main reason, and we proposed this in the SCI project. So we have two systemic fungicides, and we also have 2 biocontrol products. And I selected these treatments based on some literature review, and also especially for the systemic fungicides, they have been used in other regions, other parts of the world, but not in the US. That's why we assume, we will expect they are effective to some extent at least, right? Otherwise, the other regions of the world, the coffee growing regions would have not used those in their treatment, standard treatment. And then the two biocontrol agents, one biocontrol product, one is an extract of a plant based product, and then the other one is Pseudomonas, is a bacteria based product. These have been shown to be effective against some other, rust species or similar, fungal pathogen compared to coffee leaf rust, but not necessarily on coffee leaf rust itself. That's why we want to, I think, we think it is worthwhile to at least experimental this two biocontrol products. And both years, we have been doing this research at Corner Hills Coffee Farm. Year one, it was concurrent with our 2002 R4 coffee leaf trial. And we were doing this trial side by side concurrently, and there was only one treatment overlapping between these two treatments. That was Abound, and as you can see, for good reasons. So Abound is the next one ready to be in the pipeline in terms of label request for coffee leaf rust control in Hawaii. And year two, the R4 trial has concluded. So then there's no more R4 trial for year two, concurrent with our year two SCRI trial, but we have been doing the year two one for like 9 months already, and it will be concluded in May 2024. So this is basically the plot field layout, and as you can see, we had 6 treatments, 5 treatments plus untreated control, and 3 replications, and they are organized in a randomized complete block design. So that means that this is block 1, block 2, and block 3. Within each block, we randomized these 6 treatments, and within each plot, under each treatment, we had 6 coffee trees. And because of the area, it's relatively spatial, it's big, so we were able to actually have some real good buffer area in between any 2 adjacent plots. That means any of these, we had 6 trees in each plot, right? So we only take the data from the inner 4 trees, so that the 2 outsider trees we do not take data from, because we want to avoid or minimize the potential cross contamination between each of these 2 adjacent plots. So we did that, and here is the detailed treatment list, and also the rate of the applications, and also the application time. And I use the year 1 application time, but year 2 is basically very much the same, and we did not, like I said, it's an exact repeat of year 1, and we did not change any application frequencies, anything. So we just want to make sure, year 1 pattern and year 2 pattern are fairly similar, then we can make, solid recommendations. So 3 systemic fungicides and 2 biological control products. So the data we collected are pretty straightforward, and we are mainly collecting 2 variables. The 2 parameters. The first one is the instance of the disease. Essentially, as you can see in each replications, we use 4 trees right in the middle, and then we tagged 4 branches per tree, 2 on both sides and 2 on top and 2 at the lower section of the trees. And for those each tagged branches, and we take the 2 variables. The first variable is instance of disease. So essentially, we counted how many leaves are there on that particular twig. Branch, right? And also how many were infected with the coffee leaf rust, and then the percentage is considered the disease instance, and also the severity of the infection. So then we evaluated all the diseased leaves on each of these twigs, on each of these branches, and we gave it a rating system from 1 to 5 scale. 1 means 0 to 5 scale. 0 means no infestation. 5 means greater than 50% infestation. So then, as you can see, even at 2 rating at 2 is still relatively low infestation. But this is kind of relevant when we look at the data, when we show the results in a little bit. Yeah, and also it's important that we also look at observe the potential phytotoxicity effect on these trees, because technically these products have not been authorized to use on coffee plants in the US, right? But since we know that they have been used in many other regions of the world, especially for the systemic fungicides, and we did not expect any major, any pretty bad phytotoxicity effects on the coffee trees. Otherwise, those regions of the world would not have used this products as their treatment for the coffee leaf rust on coffee trees. So we did not expect that, and that was actually the result, that we did not observe any phytotoxicity in our year 1 project, and so far in year 2 project we have not observed any phytotoxicity result. Here I will provide some key results, and I try to probably single out the most important information I want to convey. So here, this is the year 1 from June 2022 to May 2023. As you can see, there are a bunch of treatments, but I want probably the most important message I want to convey is this treatment, which is Abound, was performing very well among all the treatments throughout the entire 1 year, as you can see here. And but that is a clear winner. To be honest, this is a clear winner, and this pattern has been held true throughout this year 1 study and also year 2 study so far. This is definitely a clear winner. However, the other 2 treatments, systemic fungicides approach and Raleigh, they were also giving some protection against coffee leaf rust for several months, for like from June to October. That's like 4 months. 4 months they were able to provide the protection against to suppress the coffee leaf rust. Then we are kind of always interested in biological control, especially for organic farms, right? Because they are they can use organic they can use biocontrol products, and then these organic products, they were doing okay, especially this howler were kind of providing relatively reasonable control for about 4 months, and then they were not as good as the systemic fungicides. But that's kind of like the overall message we saw over this more than 1.5 years in year 1 and in year 2, and also consistently in the other variables that we measured. For that is basically the coffee leaf rust severity. And if you remember, that is a 0 to 5 scale. And apparently, as you can see, it's Abound is still the best treatment among all these treatments we tested. And but all these treatments, they were all doing better compared to the untreated control. That is a given. So that means at least even if you use the products and even they do not provide longer effects against the coffee leaf rust, but they can provide some protection against the coffee leaf rust. That's a good thing, right? We can come back to this one as well. And but then again, the other 2 systemic fungicides were not as effective, or in a way that their residual effect, their treatment effect did not last as long as Abound treatment, and also they were not quite as effective as Abound in the earlier months as well. And then again, biocontrol products provided some protection, but the longevity of the protection or the treatment effect was not as long as systemic fungicides, or the effectiveness was not as great as especially Abound. That's year 1. We have entire year data. Year 2, apparently we are not done yet, but we have data until up until last month in January. And like I mentioned earlier before, my student will be going to Corner Hill Coffee Farm to take the February data next week. But again, here, as you can see, although looks kind of different because we had the entire year pattern, right? And then, but at this point, what we see here is it's very much similar to what we observed in year 1. That is, Abound is the best treatment so far among these whole treatments, among all the treatments we tested. And then the other 2 systemic fungicides, effective to some extent in the early months, but not quite as long. The longevity of the treatment effects was not as long as Abound, apparently. And the biocontrol also was okay, provided several months of protection, but not as effective or long the effect compared to the systemic fungicides, especially Abound. Similar information here. As you can see, this is the rating system, right? The average severity system, 0 to 5 scale. And then again, Abound was doing the best among all these treatments. And then the other 2 systemic fungicides, okay, few months, but not as effective. And also the longevity was not as long as Abound. And then again, biocontrol provided some treatment, some protection, but not as effective or not as long as systemic fungicide, especially Abound. So by now, as you can see, it is at this point, if this trend holds true for the rest of a few months until May 2024, right, it is really solid data to support the recommendation of Abound as the best treatments among these, the treatments we tested. And that is actually reflected in our R4 effort as well, depending on, whether Julie's lecture was already done or it will be after my lecture. But as you can see, as she mentioned, or we mentioned that Abound is one of is the next one in the pipeline that we will request a label for coffee leaf rust control in Hawaii. So as you can see, this year, the second year 1 and year 2 is not always exactly the same, but as long as the overall trend is consistent. For example, this year we had a little bit higher coffee leaf rust pressure, right? As you can see, this is above 3, at some point already. But for year 1, the severity rating was up to 2.5. But this year we had a higher overall coffee leaf rust pressure. But the key information is Abound is the best treatment that we identified, and other treatments were working, not as effective, not as long effect as Abound, and that would be the key message that we will convey throughout this 2 years research project. Lastly, I do want to mention that we went to Puerto Rico for the R4 National Education Conference about a year ago, last year, and we observed all their coffee problems, their pest, insect problems, fungal pathogen problems, and these two caught my eyes. The first one on the left side is the coffee leaf miner, and this is the insect. They kind of making tunnels inside the leaves, and according to the farmers there and also scientists over there in Puerto Rico, they consider this definitely a worse insect pest compared to coffee berry borer that we have here. The other one is probably the most, kind of scary looking pathogen I have ever seen, is this coffee berry anthracnose. We do have a coffee leaf anthracnose here in Hawaii, but we don't have coffee berry anthracnose here yet. But we definitely don't want it. And what this looks like is all these berries are covered by this grayish fungus covering the entire berries, and the coffee tree is still in the ground. So this is a terrible looking situation for a lot of quite some of their pathogen coffee trees over there. So very, very severe, very, very damaging fungal pathogen that we definitely don't want any of this. So we need to be watchful for this new these pests that are not here in Hawaii yet. I always acknowledge all my group members, even though some of them may not have contributed to this research I presented today, but they contribute or as a group members. And also, I always have this disclaimer when I give a talk on any research related projects that included any specific treatments or commercial pesticides names. Then I always have this standard disclaimer, and also acknowledgement for all the funding agencies and also other collaborators, GEMS, who did some applications for this research, and also our, the collaborator coffee farm, Kona Hills, that we have been doing this research on their farm for almost 2 years now. And that's all I have for today. And we will be here at the live Q&A session. Thank you. Okay, I'm gonna stop recording. Why is the recording? Stop share. Okay, and stop recording.